Good morning. Thank you so much for joining. My name is Xiaorui Han from Novogene, and it is a great pleasure on behalf of the Novogene team to welcome you to our new global webinar series hosted on the Genomic Research Live platform. And the Genomic Research Live is the online platform facilitated by Novogene to provide a professional and dedicated online sharing experience for life science scientists around the globe. And the platform provides a forum for leaders from around the world in the scientific, clinical, and business space to showcase their advancements and progress in their fields. The COVID-19 pandemic has led to significant changes in the way people work and communicate with each other. It is clear that the virtual and digital environment plays an even greater role than previously ever envisaged. And these unprecedented times have led to the development of a borderless scientific community whose only focus is to share knowledge and learn from one another. This aligns strongly with Novogene's mission to advance the field of genomics through sharing knowledge and cooperation. Today, it's our great privilege to introduce Dr. Hongxin Xie from Chinese Academy of Sciences, who will provide today's lecture. Dr. Xie is a PhD candidate in Professor Wei Guidu's lab of Ecological Adaptation and Conservation Biology at Institute of Zoology, Chinese Academy of Sciences. His research focuses on using genomic tools to study the evolution, conservation, and ecological adaptation of squamate reptiles. Today, Dr. Xie will speak about his work focusing on how genomic data helps to resolve the population history of Chinese crocodile lizards and provides insights into how genetic purging works in small wild populations. If you have any questions during the talk and would like to get an answer from the speaker, please leave your questions in the Q&A box. We will have some to discuss with Dr. Xie after his presentation. And from now, I will hand it over to Dr. Xie. Let's welcome him to share with us his research.
Thanks for the introduction. I'm very pleased to talk about our recent work on population genomics of crocodile lizards. And now, shall we begin? The main contents I want to share today includes a brief introduction of our focal species, the Chinese crocodile lizard, and then the reference genome we assembled, and then the main contents of our published paper, which is about the population history of crocodile lizards and the purging of deleterious alleles in its small populations. Finally, there is a brief discussion on the ongoing debate of the importance of genetic diversity and maybe some hints for future research. The Chinese crocodile lizard is a unique reptile species. It is first described in 1930, and it is the only member of its own family. The first photo here is a typical male crocodile lizard. The two most special features about it, in my own opinion, is that it's a semi-aquatic lizard and viviparous. The second picture is a habitat of crocodile lizards. And the third picture is a female crocodile lizard giving birth to a baby lizard in water. The crocodile lizard is somewhat popular in the pet market, and so poaching and habitat destruction has already driven it to the edge of extinction. Its current wild populations are small and fragmented. You can see that many studies have shown that the population of the crocodile lizards are declining. New wild populations have been found in recent years, but overall, the overall trend of its population is still not increasing, but rather declining. Currently, there's about 1,000 individuals in the wild, or maybe less. So due to its characteristics and endangered status, reference genome will be valuable for research on crocodile lizards. And now we assembled a crocodile lizard genome using PacBio long reads and assigned the context to chromosomes using high-C data. The left figure is the high-C interactome plot of crocodile lizard's chromosomes. And on the right is a chromosome features of crocodile lizard and synteny analysis between green anole lizard and the crocodile lizard. Actually, it does not give much information from this picture, but I just want to show that our genome is fairly good. We did some basic comparative analysis of crocodile genome and other squamous species. For example, we found that the origin of crocodile lizards was dated back to 100 million years ago, about in a period of Cretaceous period. It's a time when the dinosaurs were still on our planet. And we found that compared to other lizards and snakes, the crocodile lizard has a relatively slower molecular evolutionary rate, which is consistent with its morphological primitiveness. The crocodile lizard looks like a very ancient species, and we found that its evolutionary rate is relatively slow. We also did some gene family analysis, the gene family evolution and positive static genes analysis. However, this is not the main points of our work, so we will just skip it today. Now we come to the main part of our story. Here's some basic background information. In conservation biology, the small population paradigm is a very important research paradigm. It tells about a model that when a population has come to a small and fragmented, maybe due to many different kinds of reasons. But when it's small, the loss of genetic diversity and increased level of inbreeding will lead to the reduced fitness of the whole population. And then it will lead to reduce the population size. And the reduced population size will again lead to the reduced of genetic diversity. And this is finally led to extinction vortex. And this is a very well-known model. However, the genetic purging of deleterious alleles may be able to mitigate inbreeding depression. The main evidences of genetic purging are in artificial populations, such in lab populations or in breeding programs of animals or vegetables. It tells that deleterious alleles are exposed to selection when inbreeding, and so it was purged from the population. However, in natural populations, whether genetic purging works naturally in the wild is still conflicting. Recent progress in population genomics have revealed some facts, but the study results are somewhat conflicting. Let's see, the very typical research on the purging of deleterious alleles in the wild is the one on mountain gorillas. It uses R statistic, which compares the mountain gorillas deleterious alleles, that's loss of function alleles, to some populations with larger population size. And they found that the LOF mutations is relatively lower in the mountain gorilla population. Another example is from the alpine ibex. It also uses the R statistics, and they found that the populations which underwent a severe bottlenecking have very significant lower LOF mutations. However, there are other researches that have the opposite results. For example, in the study of this is in the left is a brown ear pheasant, a kind of chicken. It also uses the R statistics, and the results are opposite from the previous studies. The pheasant has a very low population size, but its LOF alleles in the wild is actually more than the larger populations. In the island force research, it uses another statistic However, it also shows that the smaller population size, the population with smaller size has larger deleterious alleles. In this case, the problem of how purging really works in the wild and what's the pattern of it merits further research. So here we aim to use the populations of crocodile lizards and collect population scale resequencing data to solve this problem. And this map shows the populations, the samples we collect. We successfully collected 4 populations. This is maybe to our best effort, the only 4 exam populations of Chinese crocodile lizards in the wild, and we collected a total of 47 individuals. From the data, we firstly want to solve a problem is that how is the phylogenetic relationships between these populations? And is there any conservation units? From the results of the phylogenetic tree and the PCR analysis and the structure analysis from admixture, we confirm that there exists 4 very genetically divergent populations of the Chinese crocodile lizards. And we found that from the relative close correlation rate analysis in MSMC, we found that the VM population, the population from Vietnam, have been fully diverged from the 3 Chinese populations about 20,000 years ago. That's about in the last glacial maximum. So our results support a subspecies of Shinisaurus crocodilus vietnamese, the Vietnamese population subspecies of the Chinese crocodile lizard. And then to solve the population demography of the crocodile lizard, because we want to know its population history. So we use 2 methods to solve its population history. The one is a PSMC that is very popularly used. We solve its population history from millions of years ago to about in the glacial maximum. And the other one is a hypothesized ABC. It solves about the population history about 20,000 years ago to the present. And we show that there's an overall trend of declining in the last million years of the population size, effective population size of the Chinese crocodile lizard is gradually declining. Especially we see from last glacial cycle, there's a decline and a bottleneck in the last glacial maximum. Then the population expanded a little, but in the recent 1,000 years, the population dramatically declined to a local minimum size in the recent years. And maybe this described as in the process of going endangered in the recent years. We also did a relentless analysis to solve the individual relationships between our samples. We did this because we know the population size is small, so it's very possible that we sample some individuals, but they are actually very close relationships, maybe siblings or parents and kids. So we need to do some statistical analysis in a foreign analysis, but these close relationships may bias our results. So we did a relentless analysis and excluded all the individuals that show possible close relationship. There's relationships stronger than 3rd-degree relationships, and we excluded about 16 individuals. And for all the other samples left, there's no close relationships between any pairs of the sample. And based on this sample, we analyze their genetic diversity and inbreeding level. Firstly, we see a figure A. FRH is a statistic that describes the inbreeding level of individual. And we see from this plot that it is very obvious that the Guangdong Maoming population is a population from China. Its inbreeding level, or the FRH is significantly lower than the other 3 populations. And from figure 3B, we did a correlation between heterozygosity as a measure of genetic diversity of an individual to the FRH, we show that the heterozygosity declines show a negative correlation with FRH. And we show that the fraction of alleles in homozygotes increases with FRH, which tells that inbreeding has already caused the declining of genetic diversity and overall genetic loads. But as the deleterious alleles are homozygotes in an inbred population, then what change have happened to its frequency? So we did another correlation with the frequency of deleterious alleles to its FRH, and we found that it showed a declining trend. That means with more intensive inbreeding, the frequency of LOF alleles, loss of function alleles, seem to declining, which shows a trend signal of purging. And if we see missense alleles, there's some alleles that are not that harmful to LOF alleles. The trend is, there's no significant trend, not declining or not increasing. It shows that the purging works differently in loss of function alleles and missense alleles. We use another popular R statistic. This statistic is to compare the differences of allele frequency between two populations. And we found that compared to Guangdong Maoming, that is the population with lower inbreeding level, the LOF allele frequency is lower in the more inbred Guangxi Daguishan and Guangdong Luokeng population, which means that the more inbred population has lower LOF mutation alleles. But for missense alleles, the difference is not that big. This is consistent with the modern Korea results. But then we want to see if the purging is really working in the crocodile lizard populations. So we did some genomic simulations to confirm our results. This simulation is a model based on the demographic history of crocodile lizards. You see our model, we modeled two population contractions of the crocodile lizards. One is about 1,000 years ago. It's about a geometric contraction from a population size, from the ancestral population size to about 1,000 individuals. And then another contraction, a second contraction, is about 100 years ago, which simulated the dramatic decline in recent years. And from 1,000 individuals to about 50 individuals. This is to a very extreme small population size. When you want to see in a simulated population, whether the deleterious alleles will be purged in this model. And the results show that our simulation results greatly confirmed with our empirical data, observed data of crocodile lizards, because the 1,000 years is the end of the first contraction, and 100 years following the second contraction, we can see that the very strongly deleterious alleles are all purged in this short period of time. But for the moderately deleterious alleles, the purging, there's no trend of purging. And in the long term, we can see the purging of very strongly deleterious alleles continues. But for strongly deleterious and moderately deleterious, if the population stays in small population size for a fairly long time, we can see there's another trend of decreasing for the deleterious alleles. We want to test whether this trend is persistent in different parameters. So we tried different carrying capacities from about 200 individuals to 25 individuals. 25 is extremely small. And we see there's a fairly consistent trend. With the population size decreases, the purging effect enhances in the initial phase. However, in the long term, when the population size is small, the overall the increasing of deleterious alleles become more severe in the long term. So we found that both the population size and duration of bottlenecks influences the process of genetic purging. Finally, from the results of our observed data and model results, we come to some conclusion. Firstly, we found that genetic purging mitigates the inbreeding depression by reducing the frequency of strong deleterious alleles. This result comes from our observed data from crocodile lizard populations, as well as the model results. And second is that both the extent of population contraction and the duration of bottlenecks influence the effectiveness of purging. This mainly comes from our simulation results. We found that when we reduce the population size of the second contraction, the purging become quicker, but in the long term, the increasing trend of deleterious alleles also become quicker for a small population size. We found that this is mainly because the decrease in genetic diversity and the fixation of some mutations contributed to the increase of deleterious alleles. And finally, our results show that on the crocodile lizards, it has fairly low genetic diversity and high level of inbreeding, but purging is working fairly good. So it shows that genetic diversity may not be precisely predict the genetic risk of crocodile lizards, and functional genetic diversity may be more important. This one comes out from our discussion and some ongoing debate on the importance of genetic diversity, which we will discuss about later. The discussion about the importance of genetic diversity comes from a very perspective article published last year. Because the significance of genetic diversity has been studied for decades in conservation biology. But last year, a perspective in PNAS, they argue that the inflated significance of neutral genetic diversity, the paper want to say that the genetic diversity will not precisely predict extinction risk of wild populations. So something about the functional diversity or the population history or the deleterious alleles will be more important. However, not soon after this perspective article has been published, there's another article published in Molecular Ecology. About several months after that, it argued with the opinion. It says that the long-standing significance of genetic diversity in conservation, which listed out decades of research from all areas of conservation biology that support the idea that genetic diversity is very important in predicting the extinction risk of populations. And another more popular article, it also published about November last year on PNAS. It again argued about the previous article that it says that the genome-wide genetic variation is very important in conservation. We know that the argument or these ideas is important, but the more important thing is some real data or evidences from case studies. And case studies actually is still emerging in recent years or months in recent time. For example, in PNAS last year, also in the fourth quarter, there is an article. The names that the genomic evidence for inbreeding, depression, and purging of deleterious alleles in Indian tigers. They investigated genetic diversity and inbreeding and how deleterious alleles are purged in Indian tigers. And it supposed idea that deleterious alleles can be greatly purged in Indian tigers. And another article is published recently. That's our work. We also the data we provided and the simulation work we studied also support the idea that genetic purging are able to reduce inbreeding depression in the wild. So maybe genetic diversity may not be a good indicator, but we also found that population history is actually very important in predicting the purging or accumulation of deleterious alleles. So the actual situation may be more complicated than we actually thought. And our work are many thanks to Professor Du Weiguo and Professor Zhou Xuming. Our job is mainly under the guidance of these two professors, and Professor Wu Zhengjun provided important samples from the crocodile lizards, and also many thanks to all the authors from our lab and Novel Gene. And this job is funded by National Natural Science Foundation of China and National Key Research and Development Program of China. And I may be talking a bit quick, so if there's any questions, please freely ask any questions. I'm very pleased to answer.
Thank you so much, Dr. For this comprehensive and in-depth lecture. And now we will take a few questions from the audiences. And the first one is from an anonymous audience that what strategies are generally adopted when sampling wild crocodile lizards and their methods to assess the representativeness of the samples?
Well, I talked about it a bit in my presentation, but I will discuss more because in the case of sampling endangered species, the problem is that because it's endangered and the population size is small, there's very like chance that we sampled two very close related samples. So the first step is that we try not to sample the same individuals in the same place. That's in the sampling procedure. But second, in the data analysis spread, when we are doing the data analysis, it's very important to do the relentless analysis. This analysis, if you did not do it, maybe in the article review process, the reviewers will ask you if you did the relentless analysis. This may not influence your results very greatly, but doing it is necessary because it will reduce some biases from your analysis. So in doing so, we excluded many individuals that are possibly close related. And actually in our case, the amount of individuals that are possibly related is actually large, but it's good. You should remove all individuals that are possibly close related. So after this analysis, the statistical significance actually did not change a lot, but it showed a more clear trend in our following analysis, the many in this part. We did two parts. We did two analyses actually. The first one we did not do the relentless analysis, but the final version we excluded all the samples, and we found that the overall trend is more clear when we excluded all the samples.
And awesome, thank you, Dr. Xie. And the second question is from Lu that the pulp size ABC can also predict millions of years of population history, and how to choose between the softwares to predict the population size.
Currently, there are many different softwares in solving population demography, and the popular one is pulp size ABC. The popular one is PSMC, and there's also MSMC and other methods. And we choose these two methods because we want to solve the history. The most important history is the most recent one because we want to see where the population size is decreasing in the crocodile lizards. So we chose to use the pulp size ABC. This method is more precisely predicting the more recent histories, many from 20,000 years to present. But PSMC is accurate and popular, but this method can only solve the histories more ancient than 20,000 years ago. But in a more recent history, it will not precisely predict it. It is illustrated in its original article. So in our study, we choose to combine these two methods to have a more broad view of the population history of crocodile lizards. And maybe if there are more precise methods ongoing, if there's ongoing in the literature, and when doing a research, you should choose the most proper one to your focal species.
Great, thank you, Dr. Xie. And last question we have from the audience is that why there was a significant reduction during the population history? It is because there's any climate change which can lead the situation, or is there any methods could be used to study the historical data?
Which can lead to, is there any method used to study? Our reconstruction of population history mainly came from our resequencing data, is the DNA data, genome data. We just estimated from our current data the overall trend in the history. But actually, there may be some real data that will describe the trend in history. Maybe some data from fossil records, or there's another way of predicting the distribution, the SDM models, species distribution models. We actually also did that. The species distribution models will describe the possible distribution of the species in the past. And we did that actually, but not presented in our final paper. We found that the species distribution model in the current situation for the crocodile lizard actually far broader than its actual distribution, which shows that actually there's many suitable habitats around, but the real population size is small. It shows that maybe human activity is a human activity that drives the extinction of this species, but not its natural adaptation to the environment. And in many cases, it's very popular to use both the SDM models and the sequence and the demographic estimation from genome data to combine these two data to analyze the main driven of extinction of the species. And there may be many literature combining the two methods. But in our current work, we did not put the SDM model because we just want to know that the population is declining. This is a very important information for the following simulation work. So we focused on the population trend, and then we did a simulation. That's why we did not put the SDM results in here, because it may be not closely related to our main results of our study. Thank you.
Awesome. Thank you, Dr. Xie. And that's so much for the Q&A sessions. We want to thanks again to Dr. Xie and our audiences for these wonderful questions and insights. And today's Genomic Research Live webinar is coming to an end, and we greatly appreciate your attention and participation. And we will provide the presentation after the webinar on demand. As always, please stay tuned for future NovoGene Genomic Research Live webinars, and we will see you next time, and take care.